Hello and welcome to the Synthetic Biology lecture series from the University of Washington. My name is Tyler and today we will be discussing the paper Multichromatic Control of Gene Expression in E. coli by Tabor, Levskaya, and Voigt, which was published in the Journal of Molecular Biology in 2011. This paper discusses the use of two separate light tunable transcription systems which are dependent on green and red light in order to dictate protein synthesis. In order for the system to function, they had to transform three separate plasmids into E. coli. This first plasmid contains the gene sequences for the green and the red sensors. Green and red. This second plasmid works as an inverter for the red sensor. And this third plasmid functions to create the chromophore protein PCB, which is necessary for both the red sensor and the green sensor to function. In the red sensor, the protein CPH8 is continually expressed. When it is expressed, it is in its phosphorylated form here. It then picks up a chromophore protein, which makes it sensitive to the 650 or red wavelength light. So when red light is shown into the system, it becomes deactivated and dephosphorylated. When far red light or 705 nanometer wavelength light is shined onto the system, it becomes reactivated, rephosphorylated, and functions to phosphorylate the OMPR protein, which attaches onto the POMPC promoter site and activates the CI protein. The reason the researchers chose to use this inverter sequence was because it didn't make much sense or much practical sense to have the system continually expressing the protein product in the case that it would under the normal red sensor. By introducing the inverter, they make the system sensitive and dependent on red light in order to produce the reporter Laxey. The green sensor responds in much of a similar way, except in that it doesn't need the inverter in order to function. CCAS is expressed in its non-active state, which then requires green light of around 535 nanometers in order to activate it. In its activated state, it is phosphorylated and increases its phosphorylation activity of phosphorylating CCAR. CCAR then works on the promoter CPCG2 in order to increase the transcription rate of the reporter Laxey. The researchers decided to use Laxey as a reporter because of its ability to produce beta-galactosidase. Beta-galactosidase can then work on the medium in order to cleave certain proteins that are within it to produce a color change, which will be shown below. Because these gene sequences are native to other species, the researchers decided that it was necessary to test whether or not the green and the red sensors were able to work in E. coli. In order to do this, they produced the data below. Ignoring Einstein for a moment, let's look at the green sensor. So they transformed E. coli cells that only had the green sensor and some cells that had the PCB chromophore and some cells that did not have the PCB chromophore. In some of the groups, they decided to shine green light on the system. When green light was shined onto the cell culture, the production of Laxey or beta-galactosidase was increased dramatically. When red light was shined onto the system, it showed Laxey production or beta-galactosidase production that was equivalent to that of the dark. When PCB was not present in the system whatsoever, the production of beta-galactosidase was decreased even more so and was seen to be on par for all light systems. So this one had red light shined on it, this one had green light shined on it, and this one had no light shined on it, yet they all produced the same very low level of beta-galactosidase. In the red sensor, we see something slightly different. As is expected, when we shine red light onto the system, we have very high production of beta-galactosidase. However, when green light was shined onto the system, there was still a moderate level of beta-galactosidase production. This is due to the transfer function of the red sensor, which I will get to in a moment. When the system was grown in dark conditions, there was the same very low level of beta-galactosidase production. And when the chromophore PCB was not present in the system at all, we had also the same amount of very low-level beta-galactosidase production. All of this information up here can be expressed much more interestingly using this data down below. By using a mask of Albert Einstein, they were able to shine different wavelengths of light onto different cultures of the system in order to see what happened. So when PCB wasn't present, as we would expect, nothing happened. No activation, no release of beta-galactosidase, and no changes of the color of the medium. When green light was shined onto the system, it's expected that the green sensor system would respond and cleave whatever was in the medium in order to cause this color change by releasing beta-galactosidase. However, when the red system was exposed to the green light, there was very little effect. When red light was shined onto the system in the green sensor, nothing happened. And when red light was shined onto the red system, we again see this imprint of Einstein's face. So as I mentioned before, it was important for the researchers to determine the transfer function, or basically how each sensor group would respond to varying wavelengths of light. So that data is shown below. So they took the green sensor and they exposed one cell culture to, say, what is this, about 425 nanometers of wavelength of light, and then about 500, 550, 600, and 650 nanometer wavelengths of light in order to figure out what the beta-galactosidase production was at the set intensity. So as you can see, the green sensor had the most response at around 525 nanometer light, which is what we would expect based on the fact that we introduced our system for the green at 535 nanometers. The red sensor is shown over here, and it's shown to actually have quite a bit of follow-through into the green wavelength, which explains the data that we had up here, where the red sensor actually responded quite a bit to green light. So they did the same sort of transfer function for the red system, in which they took the red culture, exposed it to about 425, and so on and so forth, through the spectrum at the same intensity in order to figure out what the response would be of this system. And then as you can see, at around 750, it actually deactivates the sensor. By this point, it was necessary to see how the system would respond when both of the sensors were present within the same cell population. So to do that, they produced this transfer function data shown here. So in this set, they kept the wavelength of light set, and they varied the intensities in order to produce a transfer function that showed how the system would respond to the intensities. Up above, just to contrast, they kept the intensity at the same amount, and they varied the wavelength of light to see how the system responded. And also to note, in this one, they have a group that has both of the sensors together. So they did this transfer function for just the green, and then they did the transfer function for just the red, and then they did the transfer function for both. So when they did the green, they noticed that at about 0.01 intensity of light, or watts per meter squared is the units here, at about 0.01, after that, the system pretty much saturates, which means that their only effective range for varying the protein output, and in this case it's the beta-galactosidase, which they're reading using the Miller assay, their only effective range is going to be between darkness, 0, and 0.01 watts per meter squared. Whereas in the green, because this is a green wavelength of light for this one, this is green, which is being shined onto the system at varying intensities, the red sensor actually showed about a linear response throughout the entire variation of intensities. And then when they shined red light onto the system at varying intensities, so this is red, and then once again we have our red only sensor here, our green only sensor over here, and then we have our both sensor over here. So when they shined red light onto the red sensor at varying intensities, they found also a very similar profile as was found over here on the green sensor with green light, in that after about 0.01, the system effectively saturates. So once again, their only effective control over the output is going to be between 0 and 0.01 watts per meter squared. The green sensor had this effect where as the red was increased, it actually decreased the protein output, which is what should have been expected because we know that our green sensor actually becomes deactivated in red light. So the more red light you shine on it, the less active it's going to be. And then when you put both of them together, it's actually good to notice that there's a non-linearity here. So as you can see, like if I were to add this point 24 plus like what 21, I should get like an addition of the two, in so much that 24 and 21 make 45. But we don't really see that. Same goes with over here. They don't directly add together in order to create the overall transfer function that we see in the quote-unquote both system. And that is kind of indicative of synthetic biology in that things aren't going to just perfectly add together how you would imagine them to, which is why it's necessary for synthetic biologists to create transfer functions like this in order to actually characterize the system that they're studying. So, and then the researchers had a little bit of fun with this, and they were able to produce another mask, this one of chili peppers. So it has a red area and a green area that gets light shined through it in the red spectrum and the green wavelength, or I guess the red wavelength and the green wavelength. So as you would expect, the green sensor only group would just show the green or the stem part, and it shows it in negative because it's a black and white type effect. So when beta-galactosidase is present, it produces this blackening of the media. In the red sensor, we see a similar effect. And in this case, you notice that it's a little bit lighter. The researchers did that on purpose in order to create contrast for when they did the green plus the red. So when you get the green plus the red, you end up with this overall image. And it's kind of interesting to notice this part right here, where you have the bit of green shining within the red. It just goes to show some of the resolution in this, in that it's actually like the individual cells that are responding to this input of light and not like an overall signaling between each of them. So you're probably thinking, okay, so like how is this actually applicable? Well, in current situations in synthetic biology, cells require an input of some sort of signal in order to dictate protein output. And in most situations, this is like a chemical or a protein that causes transcription. That creates a problem because you can't change the system after you've already introduced your concentration of your signal. And if you want it to change in the future, then you need some sort of degradation pathway for the molecule that you've introduced. Light is highly tunable in that, I mean, if I decide that at this point right here, I want to change the intensity in order to create more protein output, well, then I could just change it from 0.0002 to 0.0005 in order to increase the production of beta-galactosidase or whatever molecule I put in that slot where beta-galactosidase is currently. And because they have multichromatic effect, they can actually change at each point, they can change the amount of product A and product B. It just so happens that in this case they chose to use beta-galactosidase for both of them, but I could honestly put in almost any product that I want in those regions, and then individually control them based on the intensity of red and green light that I add to the system.